Okay, so in this video, I'm going to walk you through the procedure of the bacterial transformation. And so before we get started, I want to just kind of back up a little bit and tell you a little bit about what's been done up to this point. So 48 hours ago, 2 days ago, I came into the lab and I took our bottle of lyophilized or dried out E. coli. That's how it comes from the company that we order it from. And I added to that 250 microliters of this stuff, which is called LB broth. And LB stands for Loria broth. Okay, it's a nutrient media that E. coli thrive in. They grow very, very well. It has all of the nutrients that the bacteria need to live. So I added some of this to the lyophilized or dried out E. coli, and what that does is it wakes them up and it helps them to grow and to start to metabolize and be successful. So I allowed the bacteria to grow in that broth right back there in that incubator, which is at 37 degrees Celsius. That's the temperature that they like to live at. And I let them grow for about 24 hours. And so during that period of time, the E. coli kind of woke up, started doing cellular respiration, started doing normal metabolic activities, and replicated. And so what I ended up with at the end of that 24-hour period was a really concentrated vial of E. coli. Now the E. coli that we're using in here is laboratory grade E. coli, which means it's not going to cause any kind of infection for me. However, should always wear gloves, okay, as well as protective eye gear whenever handling a microorganism like that, just in case, okay? And so then yesterday, after that 24-hour incubation, I came in and I started our starter plates. And so what I did, I'll hold one up for you. I want you to take a look at this, and I'm going to hold it closer to the camera in just a second. I took E. coli from our vial. What I did is I just took a sterile loop, which I'm going to show you in a little while, and I dipped it into the E. coli, and I started to streak it on my plate. And I did it in a very specific pattern. Bring this up a little closer. The lights in here are a little bit glary. So what I started right here, ooh, camera's opposite direction, right here, if you look right here, I started to streak back and forth right here. Okay, and so you can see there's a really dense kind of lawn of bacteria right there. And then to kind of thin it out, what I did is I grabbed some of that material and I pulled it in this direction. And so you can see I made a couple more streaks there. And then I did the same thing, I pulled it and I went in this direction here. Camera's opposite of what I'm doing. And then I pulled it some more and I pulled it in this direction here. So what was I doing? I was essentially trying to thin out the bacteria. What I wanted to end up with was individual colonies. And it's still, this is not a perfect plate. Okay, I still don't do things perfectly. But if you can look, I'm gonna ask you some questions that you'll have to finish in one of the question sets here about what the starter plate of E. coli looked like. So this is our starter plate. And so take a look. What color is the E. coli? Okay, so if I'm looking here, the agar behind it's kind of a yellowy color, but the bacteria themselves are kind of like a milky white. Okay, so they're pretty white. Hopefully you can see those. And I also ask you, just to describe what the plate looks like. So you would say it's kind of a lawn of bacteria, although hopefully you can see right here, we do have some individual colonies. Can you see those? So a colony is a little circle, okay? And a colony started from one bacteria that then replicated in that place. So there's probably, there's lots of bacteria right in that little tiny circle, colony. So I ask you to describe what the colonies look like. So again, you might want to take a sketch of this plate so you can refer back to it. Colonies are pretty small. If I were to measure one, maybe a millimeter in diameter. They're pretty teeny. And my goal, what I'm going to do here is when I go to start the experiment, there's going to be a step where I have to grab a single colony. And so I'm going to try to grab one of those single colonies off this plate. I made another plate here. This one is actually worse. It's a little bit thicker. However, there is a spot. Let me try to line that up for us. The light in here is bad. Okay, there is a spot, I'm not sure if you can see it, on the plate. Let me back it up a little bit. Let me do this. That's a little bit better. Where there is some single colonies. I'm not sure how well you can see that. Okay. Again, the lighting in here is not fantastic, just like with our gel. I ended up having to take the gels home and look at them there. So again, you're going to be asked some questions, so just take a look at this plate. They won't be difficult questions, just you know, what color, describe the shape. A colony is roughly a circle, and again, they're pretty small. Actually, this plate is pretty good. I do have individual colonies on here. So again, they're small, you know, less than 1 millimeter in diameter. Okay. There's one other thing that I want to show you, and what I have to do for this is I have to turn off all the lights in here. And what I'm going to do is I'm going to hold up my little UV pen light here. Okay. Does it work? I may have to get a different one out of the back. Yeah, there it is. So this emits UV light. And the question that we're going to ask on these starter plates is, do the bacterial colonies, do they glow under UV light? Okay, so I'm going to pause it quick, and then I'm going to come back with the lights off. Okay, so I've gotten it as dark as possible in here. I know I look kind of grainy right now. But what I want to show you here on our plate, I'm going to go ahead and I'm going to shine the light on the plate. I'm not sure if you can see this or not. Okay. So do you see anything glowing green? Okay, actually, look, it looks like it might be a little bit because of the lighting in here, but I will tell you, nothing is glowing back at me green right now. So all of the bacteria on here are not capable of growth, or sorry, are not capable of glowing. Okay, so none of these bacteria have been transformed yet. None of them contain the green fluorescent gene. In fact, this is just LB media, so this is just L'Oreal broth. So we know that if the bacteria on here were glowing, then there must be arabinose, that sugar that's required to turn the GFP gene on, and that's not even in this media. So we don't have the proper nutrient, the sugar, as well as these bacteria are not transformed. They don't have the pGLO plasmid. So just normal old E. coli, they don't glow on their own. Okay? So there's no glowing on the starter plate. There is just growth of colonies that are about 1 millimeter in diameter that appear a nice white color. Okay? Okay. So the next thing I'm going to do, I'm going to go ahead and I'm going to turn the lights on, and then I'm going to go ahead and I'm going to start the experiment. Okay, so I've got the lights back on, and I'm going to go ahead and I'm going to get my gloves on. So I want to make sure that I'm doing this sterilely, right? And I'm going to go ahead and I'm going to start at my next spot on the procedure, right? So I've already made my E. coli. I grew it for 24 hours in broth. Then I plated it to create my starter plate, right, for another 24 hours. And now I'm ready to go ahead and start my transformation procedure. So I'm going to start by labeling 2 tubes. Oh, here's my foam rack. Okay, so I have my foam rack ready to roll. That's my test tube holder. And I'm going to grab 2 microcentrifuge tubes. And I'll grab a blue one and a purple one. Okay, and so now I'm going to label these tubes. And again, I'm going to label them on top with my Sharpie so that when I put them into the water bath, which I'm going to have to do in a few minutes, I don't, the marker doesn't come off. So one of my tubes I'm going to label plus pGLO, and the other one I'm going to label minus pGLO. Okay. Okay. So I've got my 2 tubes ready. Now what is pGLO? Hopefully you know, you read the introduction. And so pGLO is the plasmid. It's our vector. This is going to, this plasmid is going to hold the genes which we want to insert into our bacteria. Okay, and so what kind of genes does this plasmid hold? A couple of important genes. So the first gene that it holds is the BLA gene, the BLA gene, which codes for ampicillin resistance. So if I have the antibiotic ampicillin on my plate, only those bacteria that have been transformed will be able to grow. Okay? And then the second gene that's important is going to be the green fluorescent protein gene. Now, there's a really nice gene regulatory system built into this plasmid where the only way that the GFP gene is going to be turned on is in the presence of arabinose. So the sugar arabinose has to also be present on the plate in order for that gene to be turned on. Okay, so I have one tube where I'm going to put bacteria and the plasmid. So the bacteria that's going to end up in this tube with the plus pGLO on it are going to get the pGLO plasmid. So they're getting all those special genes, the antibiotic resistance gene and the green fluorescent protein gene. Okay, now in the other tube, the minus pGLO tube, I'm going to put bacteria in here, but I'm not going to give them the plasmid. And so these bacteria will not be able to live in the presence of the antibiotic ampicillin. And even if we feed these guys sugar, arabinose, they still won't glow, okay, because they don't have the GFP gene. Okay, so this is kind of like our control tube. We're going to treat this tube the same way. We're going to treat these bacteria the same way that we treat the bacteria that we put into the plus pGLO tube, except for, again, they're just not getting the DNA. Okay, so on to the next part. I've got to add in 250 microliters of my transforming solution into each of my tubes. So the transforming solution is called calcium chloride. And this transforming solution is going to make the bacterial cells more apt to take in external DNA. So it's going to make it easier for the plasmid to get in. Okay, so I'm going to add 250 microliters. I'm going to get myself a fresh pipette tip. And here I've got a big pipette. It's still a micropipetter, but this is set to 250 microliters. And I'm going to open up the cap on both of these. And I'm going to put 250 microliters into each of them. Okay, so 250 into my plus pGLO tube. And 250 into my minus pGLO tube. Okay, and I'm going to go ahead and I'm going to get rid of my pipette tip. And I'll put my micropipetter back over here. Okay, so now I've got transforming solution in each of my tubes. And it says to place on ice next. And I'm going to do that. So I'm going to take my rack and I'm going to go ahead and I'm going to put it right into my ice bucket. Bring the ice bucket over here to me. Okay. Okay, now it says, now it's time to get bacteria. So I'm going to take my starter plate and I'm going to take a sterile loop. And so actually, I usually use disposable loops in the lab, but because you guys aren't in here, I didn't want to open up a new pack. So I'm just going to use a metal loop. Okay, same thing. I'm going to just flame sterilize it to make sure there's nothing on here besides the bacteria that I'm going to pick up in a second. So I'm going to flame sterilize this, let it cool for a second, and then I'm going to go ahead and I'm going to pick up one colony of bacteria and put it into one of my tubes, sterilize it again, and then pick up the second colony and put it into my second tube. Okay, so I've got my Bunsen burner over here. I'm just going to flame sterilize it. Okay, let it cool nice before I touch my bacteria. I don't want to touch them with a hot loop because the heat will kill them. Okay, I'm going to just touch the agar. That nice and cool. Okay, so I'm going to go ahead and I'm going to try to pick up a single colony. I got one. Okay, literally they just wipe off the agar. It's pretty easy to pick up a colony. I'm going to pick up my plus pGLO tube first. I'm going to insert my loop into the tube all the way down to the transforming solution. And I'm going to go ahead and I'm going to twist it back and forth between my thumb and index finger. And I'm just trying to get my bacteria off. Oh, yeah, and into solution. I think that worked. Doesn't seem to be anything left on the tube. Kind of tough. Okay. A lot of times in molecular biology, we're working with, small microorganisms or microliters of things. When we do these experiments, sometimes we do this and hope for the best. I'm running my gel last week. Same thing. You load your DNA on. And throughout the experiment, you've got little tiny bits of DNA in the bottom of that tube, right? 5 microliters, 10 microliters. It's just such a small amount that oftentimes you kind of have your fingers crossed. But usually it works. Everything's on the micro scale. So again, I'm just going to touch my loop, make sure it's cool enough. It is. And I'm going to go ahead and I'm going to pick up another colony. And I'm going to put it into my minus pGLO tube. And so now, I'm just going to kind of twist it. Just shaking it in there, twisting it to make sure the bacteria come off of my loop. Because if you don't have bacteria, it's not going to work. Cells will not be transformed. Okay. So I'm going to actually take them out of the foam rack and really get them in the ice. Keep them nice and cold. Okay. And now I'm done with my loop here. I'm going to sterilize it one more time just to make sure there's nothing on there. And I'm going to go ahead and I'm going to turn off my Bunsen burner. Okay, so now in my test tubes, or my microtubes I should say, my microcentrifuge tubes, I've now got transforming buffer and I've got E. coli. Okay, so what's next? It says to go ahead and before we add pGLO to our plus pGLO tube, we want to take a look. So this has this little vial here. This has the pGLO DNA in it. Okay, and now you got to ask the question, we have to make sure that the pGLO plasmid, right, containing those genes that we're interested in, right, the ampicillin resistance gene and the green fluorescent protein gene, we want to make sure that the plasmid itself doesn't glow. It shouldn't, right, because the glowing is the result of a protein, not the DNA, and so the protein has to be first transcribed and then translated before we get the glowing of the protein. So the DNA itself should not glow. But just to make sure, we're going to take a look at it. Here's my UV light. Okay, I don't think I have to turn the lights off for this. It shouldn't glow. Okay, is there anything in here that's glowing? And the light hitting it actually on the computer camera kind of looks like it's glowing, but I promise you, nothing is glowing in here. Okay, so again, that might be one of those lead-up questions that you're going to want to answer. Okay, not only are there going to be questions about the starter plate, but there might also be a question about the plasmid DNA. So the plasmid DNA by itself, did it glow? No, doesn't glow. Okay, so the DNA itself is not the producing the glowing. Okay, remember it's going to be the protein product. So first we have to get this into our bacterial cells, and then those cells should glow. Okay, so let's go on to the next step. I'll keep that on ice. So we know that the DNA itself does not glow. Okay, so we noted our observation there. The DNA itself does not glow. Okay, now it says to, let me go down on here. Oop. I lost my protocol. Okay, here it is. So now it says to go ahead with a micropipette, we're going to add 5 microliters of that pGLO DNA to the plus pGLO tube. We'll close it and return it to ice. We're not going to add anything to the minus pGLO tube. That's our control tube. It's not getting the DNA. We're going to mix and centrifuge, and then I'm going to put them on ice for 10 minutes. Okay, so let's do this next couple of steps here. So I'm going to need to put my glove back on first, and then I'm going to need my smaller micropipette. So here we go, and we got to make sure that it's set to 5. So we're doing 5 microliters. Okay, so I just turned my dial. I got it set to 5. I'm going to go ahead and grab a pipette tip, and that's nice and sterile. Now I didn't put this down on my bench top. I put it on the edge, so the pipette tip is hanging off. I never want the pipette tip to touch anything. I want to make sure that it's sterile. And I'm actually done with my starter plate. So I'm going to go ahead and put it over off to the side. I'm going to take my plus pGLO tube. I'm going to pop that open. I'm going to take 5 microliters. I'm going to kind of flick it at the bottom. That's how I mix things. Okay, and I'm going to go ahead and I'm going to pop the top off. I'm taking 5 microliters out of my pGLO DNA solution. And 5 microliters is a pretty small amount. Okay, it's just there in the tip. And I'm going to go ahead and I'm going to add it to my plus pGLO tube. So now I have E. coli in here. Now I have my pGLO DNA. And I'm going to go ahead and I'm going to mix that a little bit. Okay. And I'm going to close the top. I'm going to flick it to stir it. I got my microcentrifuge right here, so I'm going to go ahead and add that tube. And I'm going to treat this tube the same way, right? Everything's the same except for this did not get the pGLO DNA. I'm going to do everything the same to that tube though. Okay, here comes the centrifuge. Little noise. Okay, so now I just spun this down so that again I could make sure that both my DNA as well as my bacteria are towards the bottom of those tubes and not, sitting on the side. And I'm going to go ahead and I'm going to put them back on ice. So I've got an ice bucket here with some crushed ice. That's the best way to keep things cold. It's like a slurry of ice. Okay, and now our lab protocol says to incubate for 10 minutes. Okay, so I got my timer ready. Okay, I know what I'm doing. All right, so during these 10 minutes, a couple of things I want to talk to you about. So we didn't add DNA to the minus pGLO tube. So think about why we didn't add it. Okay, so hopefully you're thinking, well, that tube has bacteria, but we're not going to give that tube the plasmid because that's our control. So if we take bacteria from that tube and we spread them out on a plate, that's what we're going to do with them eventually, right? If the plate contains just Loria broth, nothing else, those bacteria will grow, right? Just like our starter plate bacteria grew, same conditions. But if I take the bacteria from the minus pGLO tube and I put it onto a plate containing ampicillin, will it grow? Hopefully you said no, because the E. coli that we're working with are not resistant to the antibiotic ampicillin unless they've taken up the plasmid. Okay, so our bacteria lack the antibiotic resistance gene, which is called the BLA gene, the BLA gene, and so they should not grow in the presence of the antibiotic ampicillin. Okay, what if we took the minus pGLO bacteria and we grew them on a plate that has ampicillin and that has arabinose? We still won't get any growth. Okay, what if we took the minus pGLO bacteria and grew them on a plate, just an LB plate, normal broth, normal medium, no antibiotic, but we added the sugar arabinose? Would they grow? Yes. Okay, so they don't have the plasmid, but they're just grown on a plate without antibiotic. And so yes, they will still grow. So here's the second question. Will they glow? No, they shouldn't, right? Arabinose is present, but the gene, the GFP gene, is not. So you can't turn a gene on that doesn't exist in the bacteria. So no, they will not glow. Okay? Let's talk about the plus pGLO tube. These bacteria are getting the plasmid that contains all those great genes. Okay, now not every bacterial cell in that tube is going to get transformed. Only some will. Okay, only those that get transformed will be able to grow in the presence of the antibiotic. Okay, so those plates that have antibiotic on them, they're not going to look like our starter plates. They're going to have less bacteria. And that's because some of the bacteria in that tube, again, they won't pick up the plasmid, so they won't be able to grow on those plates. Okay, so let me ask you a couple more questions. So the bacteria in the plus pGLO tube, will they grow on just plates that have Loria broth? Just the LB normal media plates? Yes, they will. Will they glow on those plates? No, they won't, because arabinose won't be present on that plate. Okay? What if we took the plus pGLO bacteria and we grew them on a plate that was both LB, regular broth, regular media, and contained the antibiotic ampicillin? Will they grow? Yes, some will. Any bacterial cell that took up the plasma will grow. Will there be some that won't survive? Yes, because not all of the bacteria in that tube are going to become transformed. Okay, but those that are transformed will be able to grow. Okay, one more question on that plate. So the bacteria on the plate, the plus pGLO bacteria, those bacteria that took up the plasmid, if we plate them onto a plate that has Loria broth and the antibiotic ampicillin, will they grow? Yes. They contain the gene that codes for antibiotic resistance. So they'll be able to grow in the presence of the antibiotic. They're resistant to the antibiotic. It won't kill them. Will they glow on that plate? No, because we didn't say there was any arabinose on that plate, right? So the plus pGLO bacteria on a plate that's LB and AMP, so has lauryl broth, as well as ampicillin, they will grow, but they should not glow. We're going to do another plate that's going to contain LB, normal media, AMP, ampicillin, as well as ARA, which stands for arabinose. That's the sugar, right? The arabinose is required to turn on the GFP gene. So again, will they grow? Yes. Will they glow? Now, yes. Okay? So our plus pGLO bacteria will glow in the presence of arabinose. Okay? All right, so we still have a few minutes left. So while we're waiting, what we're going to do is we're going to get our plates ready. So I've got 2 plates here, which I put on these. I got to pop my glove off for a second. I put on these first 2 plates just lauryl broth. So it's just normal nutrient agar medium. Okay? And so on both of these plates, I'm going to write on the bottom, never the lid, always the bottom, because the lid could fall off. Okay, so on the bottom of both of these plates, I'm going to write what I'm putting on here. So on one of them, I'm going to put bacteria from the plus pGLO tube. And on the other, I'm going to put bacteria from the minus pGLO tube. So again, both of these plates just have lauryl broth, just have nutrient agar medium. There's no ampicillin, there's no antibiotic, there's no arabinose, no extra sugar. So let me ask you a question. On the minus pGLO plate, once I get bacteria on here, are they going to grow? This is from the minus pGLO tube. Yes. There's nothing on here preventing their growth. Will they glow? No. They don't have the gene, plus there's no arabinose on this plate. How about this other plate that I just labeled? Oh, yeah. This says plus pGLO on the bottom. Okay, so I'm going to put bacteria from the plus pGLO tube onto here. Will they grow? Yes. Will they glow? No. Why? There's no arabinose on this plate. Remember that sugar is required to turn the GFP gene on. Okay, so our first 2 plates are ready. Let's go to our second set. So on the second set of plates, just like I told you in the introduction, these plates have LB medium, right, so the regular nutrient broth agar, and they both have ampicillin. So now both these plates have antibiotic in them. So again, I'm going to label them. One is going to be plus pGLO. The other is going to be minus pGLO. So let's start with the minus pGLO plate first. Oh, this plate might have something else growing on it. So the minus pGLO plate, again, we've got nutrient agar, and we also have the antibiotic ampicillin. Okay? Will bacteria grow on here? No. The bacteria that I'm putting onto this plate, they do not have the plasmid, so they don't have the antibiotic resistant gene. Okay, so they should not grow on here. They're not going to glow if they don't grow. Okay, second plate. Plus pGLO tube, so these are bacteria that have been transformed. And again, on this plate, I have the antibiotic. Will they grow? Yes, only those bacteria that have been transformed. So not all the bacteria in that tube are going to grow. So we're going to have less growth on here than we did on our starter plate or on just the LB plates. But we are going to have growth. Will they glow? No. There's no arabinose on here. Okay? So there's my second set of plates all set up. We're getting close to our time. Our third set of plates. Okay, so these plates have LB plus the antibiotic ampicillin plus arabinose, which again we use ARA to stand for arabinose. Okay, so again, I'm going to make 1 plus pGLO. And I'm going to make 1 minus pGLO. We'll start with the minus pGLO first. Am I going to get growth? Hopefully by now you're saying no. Nope. These do not have the plasmid. They do not have antibiotic resistance. Okay, there's going to be no growth and obviously nothing to glow. Last plate. Last plate says plus pGLO. Will I get growth? Yes, any cell that's been transformed will grow. I'm not going to get as much growth as I got on my starter plate, but I'm going to get growth. Second question, will they glow? They should. Yeah, the sugar arabinose is here. It's in this plate. And so it will turn on, it will be used to turn on the GFP gene. And so the green fluorescent gene should be on. That protein product should be made. And if I shine a UV light onto this plate after I have bacteria, those bacteria should glow a nice, beautiful fluorescent green. Okay? So let's set it up. We're ready. Okay, so what am I going to do next? I've got to heat shock these guys. So I have my bacteria in transforming buffer, right, which is great. It's good. The cells are getting ready to be transformed. Now I've got to heat shock them. So what does that mean? I'm going to take my 2 tubes and I'm going to hold them in a water bath that's around 42 degrees Celsius for exactly 50 seconds. Okay? And after that 50 seconds is up, I'm going to pull them off quick and I'm going to get them right back into ice. So here we go. I'm going to put them in my foam rack and I'm going to walk myself over to my water bath. Check the temperature. Yeah, it's a little higher. Let me just throw a couple of ice cubes in it. Bring down the temperature just a little bit. Should come down pretty quick. And again, I'm going to heat shock them, so 50 seconds. I got the clock running. Here we go. So what's happening here is the heat shock with the transforming solution is kind of opening up the membrane around the bacterial cell, creating some of these pore-like structures, which is going to allow the plasmid to move in. So it's helping with the uptake of the foreign DNA. Got a couple more seconds left. And then we're going to pull them out and put them right back onto ice. And here, let's take a look at our next step. The ice incubation is going to be 2 minutes. Okay. And then we're going to add some broth. Okay, that's been maybe a few minutes, a few seconds more than 50. So I pulled them out of my water bath. That was my heat shock. And now I'm going to go ahead and I'm going to get them on ice for 2 minutes. Okay. And during those 2 minutes, I think I'm going to pause and get the next set of materials right in front of me. Okay, I got about 30 seconds left. But everything's set up here in front of me. I've got my micropipette set at 250 microliters. I've got my tips ready. I've got my foam block ready. And I've got my L'Oreal broth ready. I can actually take the top off that. Now again, I had one plate that looks like it got a little bit of fungus on it. Sometimes when pouring these, I don't know what happened. But it's not bacteria, so that's good. Okay, I'm ready. So I'm going to go ahead and I'm going to take each tube and I'm going to just pop the tops off, the caps. And, maybe. Okay. I'm going to go ahead and I'm going to put 250 microliters of L'Oreal broth. Again, this is just plain old nutrient. And everything that this guy requires to live. E. coli likes L'Oreal broth. Okay. Change the tip, of course, in between my tubes. And again, I want to treat my minus and pGLO tubes the same, except for the DNA. Okay, definitely controlling in this experiment. So again, the manipulated variable here is the presence or absence of that DNA. It's the DNA is the manipulated variable, right? That plasmid. Okay, so I've got them now in L'Oreal broth on my bench top, and now it says to go ahead and incubate the tubes for 10 minutes at room temperature. Okay, so during this 10-minute incubation, again, I'm going to pause and I'm going to get set up for the next step. Okay, so that incubation is over. So I just did 10 minutes at room temperature. Again, just for fun's sake, I'll go ahead and shine my UV light. There's nothing glowing in there. Okay, I'm getting a reflection off the tube, yes, but nothing is glowing. Okay, you'll see what glowing looks like once we get the results of these plates. So now it's time to plate. So I'm going to do this using sterile technique again. So I've got my metal loop here, which I'm going to use to spread my bacteria, and I'm going to flame sterilize it. Okay, so I'll get my Bunsen burner on. Maybe, yep, there we go. Okay, I'm going to go ahead and flame sterilize. Okay. I'm not going to touch any surfaces until I'm ready to roll here. And I think I'm going to plate all of my plus pGLO plates first. So I've lined them up. I've got just my plain old LB broth plus pGLO plate. I've got my LB amp, and I've got my LB amp aura. And so what I'm going to do on these plates is I'm going to add 100 microliters of my plus pGLO bacteria to each of these plates, and then I'm going to go ahead and just spread the bacteria out. And I'm not looking to create single colonies here. I'm just looking for a lawn of bacteria. Okay, so I'm going to go ahead and I'm going to get a fresh tip. I'm going to pop my tube open, and I'm going to go ahead and start. So I'm going to take 100 microliters and put it on my first plate. Again, I'm going to try not to pick the lid up too far, just so that I don't get anything else growing on these plates. Again, in the perfect situation, I would be inside of a hood right now with one-way airflow. That's really the most sterile way to do this. But we don't have that in our GB lab. And we don't do a whole lot of sterile technique in here, so it's not a huge deal. Okay, so I already flame sterilized my loop here. So now I'm just going to go ahead and I'm going to spread the bacteria out. Now I'm not going to flame sterilize in between each plate because it's the same stuff on every plate. Again, this is the bacteria that came from the plus pGLO tube. So this is the bacteria that hopefully, some of it, took up our plasmid. Okay, and so I'm going to go ahead and spread out the LB plate first. So I'm literally just kind of taking it and dragging it across the plate. Oh, I forgot my gloves. I'll wash my hands really good after. And I won't touch the plate. Okay, so there's my LB plate done. You know what, I'm going to flame sterilize just to be safe. Who knows if there was something on that plate. Now the second plate that I'm going to go ahead and spread, again, it's got my bacteria, it's plus pGLO, and this plate has ampicillin added to it. So it's our LB regular agar plus the antibiotic. Am I going to get growth? I hope so, right? Because these bacteria contain the plasmid, hopefully. So again, I'm just going to spread it out on the plate. Okay. And my last plate here, again, still has the plus pGLO bacteria. That's what I put on all three of these plates. Now this plate has L'Oreal broth, medium, plus the antibiotic ampicillin, plus arabinose, our sugar. So should these grow? Yes. Should they glow? Absolutely. This is the only plate that should glow. Okay. Okay. Spread it out. Hopefully that's cool. Okay, so those three plates are done. Flame sterilize. Now, I'm going to move those plates over so I don't get confused. I have my other set of plates, again, an LB ampicillin, and an LB ampicillin aura. And I'm going to go ahead and spread these out, hopefully in that order. Okay, and I'm going to take my minus pGLO tube, pop it open, and again, I'm going to put 100 microliters of the bacteria that have not been transformed. There is no plasmid. I'm going to go ahead and I'm going to get them on these plates. So as I'm doing this, tell me, which plate am I going to get growth on? There's only one plate. I should only get growth on my first plate, the LB amp plate. plate. Sorry, the LB plate. I was opening the LB amp plate. So these bacteria do not have the plasmid, and so therefore they should not grow in the presence of the antibiotic ampicillin. Okay, and so I like a clean workstation. So the only plate here that I should get any growth on is the LB plate, because the other two plates have ampicillin. Okay, and the plate where I get growth, I shouldn't get any glowing because there's no plasmid, and there's also no arabinose, the sugar, required to turn on the GFP gene. Okay, what a cool lab. We're learning about transformation. We're also learning about gene regulation. Okay, lots of different stuff all in one. Love biotechnology. And of course, in this lab, this is just for fun, right? We're just putting a glowing gene into our bacteria just for fun. But remember, there are lots of real-world applications for transformation, right? Think of all the types of genetic modifications that are being done. All of those are really, really important. Okay. Oh, my computer's talking to me. Hold on. Okay. Thought I was almost out of storage there. Okay, so I'm going to go ahead and spread out my next one. Again, this is trying to just avoid, there was a spot that had some, looks like a fungus got in here. That's the problem with not using a hood, one-way airflow hood. Sometimes you get some growth of organisms that don't belong on your plate. Try to do this as sterilely as possible. I haven't even opened this plate yet. All I did was pour media into it when I was making it, and looks like it got a little something. So here's my last plate, so I'm going to go ahead and spread it. Again, this has E. coli that is minus pGLO, so it does not have the plasmid. And it's being grown on a plate that has LB broth plus the antibiotic ampicillin plus the sugar arabinose. But because there is no plasmid here, we should not get any growth and therefore no glowing. So that's it. Those are my 6 plates that I'm going to make. Okay. Now what I'm going to do is I'm going to tape these up. I'm going to turn them upside down because we get better growth when plates are turned upside down like that. And then I'm going to put them back in my 37-degree incubator back there in the back of the room. And I'm going to leave them overnight. And I'm going to come back tomorrow and I'm going to make another video showing the results of this. Okay? So stay tuned.